This is a neat paper on long COVID exercise and post-exertional malaise, where they show that skeletal muscle structure is associated with lower exercise capacity in patients, where this is caused by local and systemic metabolic disturbances, in addition to tissue infiltration within the skeletal muscle of patients with long COVID. In this study, they systematically induced post-exertional malaise in a cohort of 25 well-defined patients with long COVID and controls, where they obtained blood from the patients and did skeletal muscle biopsies before and after a maximal exercise test. Results were then compared with 21 age and sex-matched controls, where those controls were patients that fully recovered from a mild COVID infection. Prior to COVID infection, both groups were healthy and socially active, and none of these patients were hospitalized due to COVID infection. So to reemphasize these patients that have long COVID and this post-exertional malaise, it was all from a mild COVID infection. These were not hospitalized patients. The first finding was that the maximal oxygen uptake, or the VO2 max, was substantially lower in long COVID patients, as was the peak power output. Patients with long COVID also had lower maximal ventilation and lower maximal end tidal partial pressure of CO2, which also implies poorer ventilatory function during exercise. As an aside, in these long COVID patients, the cardiovascular system was not compromised, so the authors do not believe that that is why they have limited exercise capacity. Additionally, the capillary density ratios were not different between groups, and when looking at the metabolic markers for succinate dehydrogenase, SDH activity, which is a marker for mitochondrial content, it correlated well with the VO2 max in healthy controls, but not within patients. The patients had a significantly lower oxidative phosphorylation capacity, a lower ability to produce energy. And then when looking at the skeletal muscle biopsies for the vastus lateralis muscle, this part of the quadricep right here, both before and 1 day after the induction of the post-exertional malaise, they found significantly more amyloid deposits in the long COVID groups than they did in the healthy controls. So for instance, the cell membrane is blue, the amyloid deposits are green. In our healthy controls, we don't really see many. There's maybe 1 or 2. But then when we look at the long COVID group, we see a huge number, and then this would be the before, this would be the after. We see many more in the after for these long COVID groups, where these amyloid deposits are really bad because they're basically twisted proteins, and they're a direct sign of muscle and tissue damage. What's really interesting is that these cellular deposits are located in the periphery, the membrane of the cell, as opposed to within the cell nuclei, which suggests that these deposits are located outside the infiltrating immune cells. So the authors conclude that these amyloid-containing deposits are not present within the actual capillaries themselves. So they don't think that post-exertional malaise can be explained by these deposits because they're not blocking vessel perfusion. However, what they are seeing is a consistent reduction in parameters that we mentioned earlier, such as the VO2 max, the peak power output, the oxygen-gas exchange with carbon dioxide. And these trends are pretty consistent between the long COVID and healthy groups, the long COVID being the red, the healthy being the white. However, they are seeing that the healthy controls are maintaining at their energy conversion levels, but people with long COVID, the patients, are constantly having a reduced maximal mitochondrial respiration and output. So while we're seeing that they're either staying the same for the healthy group or actually improving in terms of that mitochondrial efficiency, that energy activation, people with long COVID are having consistent reduction in those parameters. Additionally, these long COVID patients are displaying small atrophic fibers and focal necrosis, which means that the cells are showing death and decay, which means, in other words, that exercise is exacerbating tissue damage in these patients, where this is a demonstration of that atrophy within the muscles, and then this is the demonstration of that necrosis within the muscle tissue, which is significantly higher in that patient long COVID group being the red, with the healthy control being the white. Additionally, in the long COVID patients, there's a higher level of immune response with these CD3 cells and these CD68 cells at both baseline and 1 day after. So these CD68 macrophages and the CD3 T cells suggest a locally disturbed immune response in patients with long COVID. So in that skeletal muscle, you're going to have both local inflammation from the hyperactive immune response, and you're going to have all these tiny little deposits in the surrounding structure of the fibrous network. and you're going to have a low cellular metabolism, which combine together to create muscle pain, fatigue, and weakness, where the muscle pain is coming from both those tiny little, essentially crystals in the tissue, which is making it hard to move without having some sort of obstruction, in addition to the local inflammation, which is also resulting in some local cell death, which is going to result in pain. And the loss of that tissue from the atrophy and the necrosis is going to result in that weakness, where the fatigue is going to come into play with the lowered cellular metabolism. And interestingly, accelerometer data indicated that long COVID patients were not bedridden. They had an average step count of about 4,000 steps per day, and that physical inactivity itself does not cause post exertional malaise,